So now that we have talked all about protein structure, we talked about what proteins are, now we're going to talk about what proteins do. So this first chapter is just a generic introduction to protein function, and then in the next chapter we'll talk all about enzymes, which are a special type of protein. But pretty much all proteins work one way or another by binding to something else, some other molecule, and the something else we usually call a ligand. So the ligand can be pretty much anything. It can be a small molecule, it can be another protein, it could be another macromolecule, and that binding is the first step in carrying out whatever the protein's function is. So if it's an enzyme, for example, then we actually call the ligand a substrate, and then the function of the enzyme then is to turn the substrate into something else or catalyze a chemical reaction in which the substrate is the first step. So other proteins, though, simply bind to their ligand or bind to another protein, and then just by that binding, that initiates their function. So, and then, of course, if to bind to a ligand, the protein also has to have a binding site. So the binding site of a protein is just the location on the surface of the protein where the ligand fits. And so usually the binding site has a shape that's complementary to the ligand's shape in one way or another, and it's specific to that ligand. So most proteins have very specific binding sites that only bind to a subset of molecules, and that is, again, important for their function. And so most proteins then and they can have more than one binding site. You could have one protein with multiple binding sites, but typically either, especially if it's a single subunit protein, the other binding sites will be for other ligands. So you can have a protein that binds to multiple ligands, but each ligand would bind to its own binding site. And of course, there are cases where some proteins have are pickier about the ligands they bind to. It just depends on the protein. But protein and ligand binding is kind of like any other chemical reaction. It is reversible, so that means that even if the protein and the ligand bind each other very tightly, there's always going to be a reverse of that reaction where the ligand and the protein dissociate. So when we talk about protein binding, protein and ligand coming together, that's often called association. And then the separation of the protein and the ligand we call dissociation. And so each of those processes has a rate constant, because again they're just like any other chemical reaction. So the association rate constant, little ka, just describes how quickly the protein and ligand, when they're mixed together, form the protein ligand complex. And then the dissociation constant, little k sub d, is just the rate constant associated with the reverse, the dissociation of the protein and the ligand. And again, like any other reaction, these the protein ligand binding also have an equilibrium constant. So there's an equilibrium constant associated with the association of the protein and the ligand, and there's a equilibrium constant for the dissociation. And those use the big K, big letter K, to represent those values. And this down here, this is the relationship between the equilibrium constant and the rate constant. So the association equilibrium constant is just the ratio of the association equilibrium constant is the ratio of the association rate constant over the dissociation rate constant. And of course, it equals the concentration of protein ligand complex. So the concentration of the two molecules bound together divided by the product of the unbound protein times the unbound ligand concentration. And of course, that is the inverse of the dissociation equilibrium constant. So another way to talk about ligand binding, so if you want to know how much of a protein is actually bound, we use the term binding fraction, and the letter Y is used to represent that. And it's simply the percentage of proteins in a mixture that are actually bound to ligand. And it's a pretty simple way to calculate it is just you take the concentration of the protein-ligand complex divided by the total amount of protein in your solution, which is just the bound protein plus the free protein. And so that's what that big P is, just free protein. And then we can describe, now it's hard to measure sometimes the protein-ligand concentration directly, so it's convenient to describe the binding fraction in terms of the free ligand concentration, because that's experimentally usually easier to measure. So we can rearrange this equation to just be in terms of the protein ligand, just in terms of the ligand concentration. So if you remember from up here, we said that the dissociation equilibrium constant is just the inverse of the association equilibrium constant, which is this ratio. And so we can just rearrange that down here and say that K, big K sub D equals the product of the free ligand concentration times free protein concentration over the protein ligand complex concentration. So we can rearrange that in terms of the protein ligand concentration. So just move that over here and say that this equals this, the product again of our ligand concentration times the protein concentration, free protein concentration, over the dissociation equilibrium constant. And so plugging that into our equation over here, we can say that Y then equals this ratio. So we can say it's plug this whole value into this equation over here, and you end up with equation. I'm not going to draw the whole thing out, but we can take out some of the terms from that equation. The protein concentration disappears because you've got that showing up on both the top and the bottom. So you end up with something that looks like this. So it ends up being just the ligand concentration over that association constant, and then that same ratio down here, plus the plus 1. And then that just rearranges into this. So you have free ligand concentration over the sum of the ligand concentration plus that dissociation constant. So the dissociation constant, even though we're usually interested in the binding of the protein, the association of the protein and the ligand, oftentimes the dissociation constant is used to actually describe the affinity of the protein, simply because it's easier to measure. And so for a typical protein, you can see that the relationship between Y and the concentration is ligand concentration is a function of that, or is related to the dissociation constant. So this is a typical binding curve. So Y is always going to be a number between 0 and 1. 1 means 100% of the protein is bound by ligand. 0 means that none of it is bound. And then again, down here is just a concentration of the ligand, whatever that happens to be. And you can see that when the ligand concentration equals the dissociation constant, dissociation equilibrium constant, that is by definition, according to our equation, that will be when Y equals 1/2, because then we can just plug in over here KD. So if we say that ligand concentration equals dissociation constant, then Y just becomes this. So it's just these values added up, and that becomes 1/2. So another way to think of the dissociation constant is the concentration of ligand when the protein is 50% bound. So when 50% of the protein in your solution is bound by the ligand, then by definition the ligand concentration is equal to the dissociation equilibrium constant. Now, for some ligands, if you have, especially if you're talking about a gas, it's easier to use partial pressure instead of concentration. So we're going to be talking about hemoglobin and myoglobin in a minute. Those are proteins that bind to oxygen. Oxygen's a gas, and so it's easier to measure and talk about gases in terms of the partial pressure rather than concentration. So a lot of times you'll see this binding curve drawn this way, where instead of concentration of oxygen, we use the partial pressure. So this little p in this case means the partial pressure of oxygen gas, and then instead of the dissociation constant, you have this term called the P50, but it means the same thing. It's the partial pressure of oxygen at which 50% of your oxygen binding protein is bound to oxygen. And so it's the same basic concept. So again, we use the dissociation constant to describe the binding affinity of a protein. So affinity in this case just means how much does the protein want to bind to the ligand, how tightly do they bind together. And so you can kind of see here a general sense of what high and low binding affinities are. So KD is what we use, and it's usually given in molar. And you can see that for most proteins, actually, they have relatively medium to low affinity for their ligand. So that means that a KD of around 10 to the minus 8th, 10 to the minus 10th molar is pretty typical for a protein. So you can see here, if a protein has a low affinity for its ligand, then it has a high KD, because the KD is the dissociation equilibrium constant. So if something has a high dissociation constant, that means that at equilibrium, more of the protein is going to be unbound to the ligand. So again, for typical proteins, the average KD is in around 10 to the minus 8th or 9th. Very high affinity proteins have very low KDs, so down to 10 nanomolar would be a very tightly bound protein, whereas a weakly binding or a low affinity protein would have a KD upwards of 10 micromolar. So there's a big range. It depends on the function of the protein. So some proteins, like enzymes, for example, actually have very low affinities for their substrates. And again, substrate is just the special term we use for the ligands that bind to enzymes. And so that's important because the enzyme is not supposed to bind to the substrate for very long, as we'll learn later. The job of the enzyme is to bind to the substrate very transiently, so just for a short period of time, and then convert it into something else. So if an enzyme had a very high affinity for its substrate, it actually wouldn't be very good as an enzyme. It wouldn't be able to modify the substrate into something else. And then other proteins like receptors. Receptors are proteins in the membrane that bind to something that comes usually from outside the cell and then acts as some sort of signaling mechanism to the inside of the cell. But typically, and so these are useful for communication between cells. And so again, there you want the ligand to bind to the receptor just long enough to activate the receptor, but then to let go so that the signal can be turned off. That makes it more useful as a communication system. So those receptors have kind of medium binding affinities for their ligands. Meanwhile, things like antibodies, we'll talk about antibodies later on. Those are special proteins that bind to antigens, which can be anything, but typically hopefully are viruses or bacteria, parts of bacteria, or any sort of foreign molecule in the body. And they need to have a high affinity so that they can act as markers so that the immune system can destroy the invader. Meanwhile, there are lots of proteins that bind things like DNA, and most of them have to be sequence specific. So in other words, you don't want a protein that binds to just any DNA segment, usually, especially if it's a transcription factor, a protein that's going to control when and where other genes get turned on or off. So if you want specific, sequence-specific protein binding to DNA, then it has to have kind of moderate affinity, because you want the protein to bind to other DNA segments to be able to sort of read along the DNA molecule and look for its sequence-specific target. And so that's why it has that medium level. And then there are some proteins like biotin. Biotin is a protein that binds very, very tightly to its ligand called avidin, and those are for very specific kinds of interaction. So again, typically most proteins work by binding to their ligand only temporarily, mostly because, and we'll talk about this more later, but typically the interaction between a protein and a ligand also is non-covalent. So proteins that bind to their ligands interact with them through hydrophobic interactions and ionic interactions and hydrogen bonds, but rarely, or at least not rarely permanently, do proteins bind to their ligands covalently. So that's the other reason why affinities tend to be kind of medium or moderate for most proteins. And one last thing to say about protein binding in general before we get into some specific examples is that usually when a protein and a ligand bind, the protein at least, and usually both the protein and the ligand, will actually change shape. This is called induced fit. And again, this is important. For one thing, the induced fit usually increases the affinity. So when the protein is bound to the ligand, it takes on a conformation that has a higher affinity for the ligand than when the protein is not bound to the ligand. Also, the induced fit is what leads to the protein actually doing whatever its job is. So this is especially true for enzymes. So whenever an enzyme binds to its substrate, the enzyme changes shape to fit the substrate, but in particular, the fit changes shape in order to carry out the reaction. And often the induced fit doesn't just apply to the binding site. The induced fit means that the entire protein can change shape. And it doesn't have to be a drastic change. It's just enough of a change that it allows the protein then to do something else, maybe to bind to some other protein or release some other ligand that may have bound, or again, maybe to kick off an enzymatic reaction. So next time we will talk about myoglobin and hemoglobin as specific examples of protein binding.